In this section, I want to briefly look at a laboratory technique which is frequently used in medicine and forensic science and show how it can be used to distinguish between males and females. What I'm referring to here is the polymerase chain reaction, which is more well known by the acronym PCR. Now, the reason that so many people are familiar with that acronym PCR is because in recent years, millions of PCR tests have been performed worldwide to determine whether individuals have been infected with a particular virus. You know the virus I'm referring to. However, PCR has much broader application than just detecting a virus. Generally speaking, PCR is a technique whereby researchers can amplify many copies of a particular DNA sequence. And when I say many copies, I mean millions or billions of copies of a sequence. And when you have lots and lots of copies of a DNA sequence, such as with PCR, it makes it much easier to determine the presence or absence of a particular sequence. So how can this be used to distinguish between males and females? Well, I first need you to consider two different genes. One of them is found on the X chromosome and one of them is found on the Y chromosome. Now, these genes are very similar to each other and they are referred to as the amelogenin gene. Now, the amelogenin gene is actually a gene associated with tooth enamel. However, what the gene encodes is not really that important. What is important is that the amelogenin gene found on the X chromosome is a little bit different from that on the Y chromosome. It's actually 6 DNA letters shorter. This is how one professor of genetics describes the situation. The amelogenin gene, which produces a tooth enamel protein, is present on both X and Y chromosomes, but the X chromosome version is 6 bases shorter, and this difference can be detected by PCR analysis. Okay, so I'm going to come back to later the rest of that quote, but before I do elaborate on it more, I want you to consider two separate scenarios. The first scenario is, imagine someone commits a crime, and in committing this crime, they actually break into a building and they break a window to enter the building, and in doing so, they actually cut themselves on the glass and they leave a blood sample at the crime scene. So, can we use PCR and the amelogenin gene to tell whether the blood sample comes from a male or female? The answer is that we actually can, and I'll explain more how that's possible in a moment. The second scenario is, imagine that archaeologists are digging up human remains and they stumble across a fragmentary piece of bone, and they're not sure whether this bone comes from a male or female. So the question arises, can we use PCR to determine whether the individual was male or female? Now, the answer is yes, we can, provided that the DNA isn't too degraded. And so let's review our two scenarios. On the one hand, we've got the blood sample at a crime scene, and in the second example, we have archaeologists with a fragmentary piece of bone. Now, one other thing I might say about the fragmentary piece of bone, if the archaeologists dug up, for instance, an entire pelvis, it might be a little bit more obvious whether the individual was male or female, because males and females have distinctive differences in the shape of the pelvis. But let's imagine the fragmentary piece of bone is like a small part of the tibia or something like that. Okay, so there are our two scenarios. Let's go back to this quote from the geneticist that explains how this test can be utilised in these scenarios. The amelogenin gene, which produces a tooth enamel protein, is present on both X and Y chromosomes, but the X chromosome version is 6 bases shorter, and this difference can be detected by PCR analysis. When a DNA sample from an unknown donor or suspect is tested for the amelogenin locus, the results for a male who carries both Y and X chromosomes shows 2 separated bands or peaks, and just 1 band for females with only X chromosomes. So you see, if your PCR tests manage to amplify both versions of the amelogenin gene, the version on the X chromosome and the version on the Y chromosome, you obviously have an individual who's genetically male, XY. Alternatively, if your PCR tests only manage to amplify the version of the amelogenin gene that's found on the X chromosome, you obviously have an individual who's female because no Y chromosome is present. Now, this test would presumably also work for some of the sex chromosome variations we've already encountered. So, for instance, if you had a female with Turner syndrome who has one X chromosome, or if you had a female with triple X syndrome, 3X chromosomes, and you applied the PCR test in these scenarios, you would still only get the version of the amelogenin gene which is found on X chromosomes. Alternatively, if there was a male with Klinefelter syndrome XXY or a male with Jacob syndrome XYY and the PCR tests were applied in this scenario, once again you would amplify two different versions of the amelogenin gene, the version on the X chromosome and the version on the Y chromosome. Now, at this point, some people might say, okay, fair enough, this PCR test can inform us about the presence of sex chromosomes and tell us about biological sex, but they might say, but that doesn't necessarily tell us about someone's gender. Many people, of course, see a very sharp distinction between biological sex and gender. However, what I find particularly fascinating about this testing for the amelogenin gene, is that there are a number of papers in academic journals when discussing this gene and this test, they'll actually refer to how this is used in gender determination, and they specifically use the word gender in the titles of their papers. Here's 4 examples. Okay, with example number 1, is the amelogenin gene reliable for gender identification in forensic casework and prenatal diagnosis? How about this one? An investigation of sequence deletions of amelogenin, a Y chromosome locus commonly used for gender determination. Here's another paper, amelogenin gene, the pioneer in gender determination from forensic dental samples. And finally, this paper here, detection and analysis of null alleles of amelogenin in gender identification. So in each of these 4 papers, they are discussing issues associated with utilising the amelogenin gene, but they are referring to this gene and what it reveals about sex chromosomes as something that can be used in gender identification and gender determination. And they specifically use those terms in the titles of the papers. This just goes to show that many people still use the terms gender and biological sex synonymously. That's obviously what these researchers are doing. Now, of course, words change over time and the meanings of words change over time too. And some people will point out that the word gender isn't just based on biological features, but also encompasses things like cultural and societal roles for individuals, and I can appreciate that. However, this presentation isn't about how words change over time, and this presentation isn't about societal and cultural roles for men and women either. This presentation is about genetics and how a greater knowledge of genetics can give us a better understanding of biological reality. And what we know about this PCR test I've briefly described here is that regardless of the way an individual perceives their identity, if researchers were to examine a sample of their blood, those researchers, simply by applying this PCR test, those researchers could say with a very high level of confidence that this individual is male or female. Alternatively, archaeologists could study their bones hundreds of years from now, and simply by applying this PCR test, those archaeologists could say with a very high level of confidence that this individual who was living was male or female. And if the evidence presented by our genes and chromosomes is important, and I believe it is, then we will have far more fruitful discussions on related topics if we remember to keep this genetic evidence in mind. So in this section, we have seen how we can use PCR to distinguish between males and females, but in the next section, I want to take a closer look at some of the important changes that occur in early in utero development.